Right, welcome to Module 9. So this module is going to be on the muscular system. Here are your learning objectives as usual. So the first thing we'll do is just introduce muscles and muscle tissue. Muscle is not hard, but it is organized in such a particular way. It's a bit trippy how you have these bigger filaments and smaller ones and smaller ones and smaller ones, and we're going to try to dissect that today. So I want us to recall a little bit what we talked about in the tissues section. Recall the three basic muscle tissues that we had talked about. We talked about skeletal muscle. We talked about cardiac muscle and smooth muscles. Those were the three types of muscle tissue. For skeletal muscle, let's talk about its location. These are attached to bones or for some facial muscles to skin, but it's mostly bone to bone, right? Over here you have a bone, and here you have a bone, and this muscle here, the biceps muscle, is attaching this bone to this one, and as it contracts, it could pull on this bone, and it could really change the angle of your arm, right? It allows movement. So this is bone to bone attachment, and it's for moving, right? Moving, bones don't contract, muscles contract, and they move the bones. So that's a skeletal muscle because it's on the skeleton, it's on the bones, and it moves the bones around. And if we look at its histology, its tissue under the microscope, you will see these single, very long cylinders, right, that are multinucleated. You see multiple nuclei on the peripheries of these cells. And you see obvious striations, right? Do you see these zebra stripes? Remember we said it was the actin and myosin myofilaments that were all in line. We're going to talk a bit more about that today. Cardiac muscle, where would that be? It would be in the walls of the heart, right? All of this stuff here, here as well. This is just fat and some little arteries, but all of this, the major component of the heart is cardiac muscle, right? It's this muscle that contracts, because you know the heart, it beats and it squeezes, right? So if you look at that under the microscope, you'll see these branching chains. Do you see? Remember that these are the ones that branch out. You'll see they're uninucleate. Each of the cells has one nuclei. And also you'll see striations, right? And remember, for cardiac muscle, we looked at the Z line. Do you see these darker, sorry, not the Z line, for cardiac muscle, we looked at the intercalated discs over here. This is the region where two muscle cells will come together. So here it's full of gap junctions so that they can communicate from one cell to another. It really allows there to be a border in our cardiac cells, okay? Over here, you have smooth muscle. This one is, this is a unitary muscle in the walls of hollow visceral organs. So, other than the heart, right? So, an example of this is like in the digestive system, right? Throughout the esophagus, down the stomach to the intestines, this esophagus is continuously contracting and pushing food down via peristalsis. The stomach is squeezing and mixing up food. The intestines are squeezing. So that the outer part of those organs are all lined with smooth muscle that contract in unison to push things through. Remember, under the microscope, these are single spindle shaped, right? They're like spiny on each end, and they're uninucleate. They have one nuclei in the middle, no striations. This doesn't mean they don't have actin and myosin. It doesn't mean they don't have these myofilaments, but they're not organized in a nice way such that you see these nice striations over here. So you don't see the striations in the smooth muscle because they're all intertwined and they're not organized into nice rows of dark and light bands, okay? So I'm gonna just name a few characteristics. All muscles share four main characteristics. All muscles, no matter whether you're skeletal, cardiac, or smooth, right? All of them are excitable. Okay, they respond. Um, Muscle on its own, right, um, will not contract. If you just put a muscle on a table, it will not contract. You need to send a nervous signal to it. You need to send some kind of signal to tell it to contract. So, but all of them are excitable, right? If a nervous signal, bzz, bzz, little neuron goes to it, um, this will allow it, it'll receive that signal, that nervous signal, and respond to that stimuli, okay? All of these are excitable. This muscle here, right, is skeletal muscle. That signal comes from the brain, that neuron comes down, and it goes on to the muscle and tells it to contract. In the heart, actually, there are some nervous areas in the heart that will, um, nervous tissue, but that'll self contract the heart, which is interesting. But also, you have some neurons that help uh, to tell the heart how fast to beat. So that's bzz, bzz, nervous signals. And same thing with the stomach and the esophagus and all of this smooth muscle that contracts these tubes in their body. All of those, right, they receive nervous signals. You know that when you're hungry, right, and you see food or you think of food, sometimes your nervous system will start contracting your stomach and you'll hear there's in the middle of class. That's an example of, your nervous signals going down to this smooth muscle and it's starting to contract your stomach already. So, excitability. All three of these muscles are excitable and they respond to different stimuli and they receive outputs to contract, right? So, the nervous system does send some signals to it so that it can contract. So, excitability, it has the ability to receive and respond to stimuli. Contractibility, right? What is the response of a muscle? The response of a muscle is to actually contract, right? The ability to shorten forcibly when stimulated. All of these muscles shorten when stimulated. This long cylinder shortens. The biceps muscle here, when it contracts, right, it actually gets smaller. That's why for the biceps you see this bulge that comes out when you contract your biceps, right? That's because it shortens and gets smaller, and the muscle fibers kind of bulk up in the middle. Same thing for the cardiac muscle. The fibers there, these branched fibers, they contract and they squeeze the ventricle, right? Just like a water balloon, if you were to squeeze it, that would squeeze the liquid inside. Well, that's what the heart does, it contracts. And lastly, the smooth muscle, right? As you're actually gonna, these little spindle-like smooth muscle cells, they will shorten and they will all squeeze and make sure that the tubes and the stomach, let's say, they get smaller, right? They constrict. They're getting smaller and they're contracting too. So that's contractibility. So all muscles, they're excitable, they're contractile, they're extensible, meaning they can stretch, right? They have this ability to be stretched out, and also they're elastic. They once you let go, they have this ability to recoil back and to resist lengthening if you're pulling a muscle too much, right? It's good that a muscle is slightly elastic and it won't just snap, or else we really injure our muscles often. It happens, but we have mechanisms in place to protect us most of the time. What is the function of the muscular system in general, right? The muscular system is there for movement. So skeletal muscle is, for example, is voluntarily controlled and it contracts to pull tendons to move our bones. So here's the biceps muscle, for instance, and it has a little, the muscle goes and has a little tendon over here, a tendon over here, and as it shortens, right, the muscle shortens, it pulls here, it's pulling the bone up, right? So it allows us to move. Some muscles, a lot of them do allow us to move, and this is an example of a skeletal muscle that allows us to move. Posture, right? Continuous muscle contraction keeps our body upright by maintaining our posture and body position. It's not always the best posture, but this is one of the functions. You don't even realize it right now, but just sitting or sleeping or whatever, your muscles are contracted. It's called a muscle tone. There's a specific basal muscle tone that your muscles are constantly contracting, right? If you're a bit more tense, maybe they're contracting stronger. But right now, to keep you upright and sitting, right, your muscles are contracting. These trapezius, for example, in your back, they're always contracting to keep you upright. If not, you'd collapse, right? You know that if you faint, you collapse down. Your muscles are not keeping you up. So some muscles are there for posture. Some muscles are there for protection. Some bones surround and hold certain visceral organs, while some muscles support and protect the soft tissue. We talked about how bones protect sometimes, like the skull or the sternum protecting the heart, the breastbone, or the ribs protecting the lungs, but the muscles also do the same. Like this layer over here of the rectus abdominis, right? The abdominal muscles over here, they're actually a layer of soft tissue protection. They protect, they're really protecting this area, the visceral organs that are over here, right? They're a layer of protection. They're not doing, like a bone. They're not protecting as the bones do, but this is one of their functions. And heat production, right? Muscle contraction uses a lot of ATP, which releases some energy as heat. So, this is something that maybe I didn't mention, but when you undergo metabolism, you use oxygen, you use glucose, and you make ATP, right? This is something that muscles are one of the organs that do this the most because they constantly need ATP. Every contraction needs ATP. And if you think of this muscle that's constantly contracting throughout the day, then you're using up a lot of energy just for your muscles. Just sitting here right now, you're using up energy, which is sad. So one of the byproducts of cellular metabolism is also heat. When you burn a lot, you know this. When you go for a run, you get very warm because your muscles, your leg muscles, and your whatever muscles you're using as you're running are burning and using up a lot of ATP, making a lot of ATP, and you're constantly making heat, right? So that is important. So muscle contraction uses a lot of ATP, which releases some energy as heat. So the heat generation maintains our body temperature. I'll give you an example. Actually, our body uses this to our advantage when we're in cold temperatures. Remember I told you for homeostasis, when we're cold, we don't, we want to keep our internal body temperature stable, so we'll start shivering. Those are our muscles making mini contractions and using up a lot of ATP to produce heat so that we can maintain our body temperature, right? So that's interesting that our body does that to help us. All right, so a quick, just a side topic, just to make sure that we understand this. Skeletal muscles, we'll start with. We'll talk about their nerve and blood supply. So each muscle receives nerves, arteries, and veins. So there are neurons, for skeletal muscle, there are neurons that go to these muscles. Why? Because skeletal muscles are under voluntary control. We tell our muscle what to do, when to contract. If I ask you right now to reach for the cup on the side, your brain is telling your muscle to do that. So you need to send these yellow little nerve cells, neurons, down to your muscle to excite them and allow them to contract, right? Arteries and veins, all your muscles need constant oxygen and fuel, glucose, fat, whatever it may be, to burn and make ATP, right? So this is very crucial. So this is consciously controlled. Consciously controlled skeletal muscles, right? They have nerves supplying every single muscle fiber. Every single muscle cell has a neuron that goes down to it. That's how important it is to send these nerves. Um, Contracting muscle fibers, right, require huge amounts, as I said, of oxygen and nutrients, and they also need waste products to be removed quickly. All of these cells in this muscle here, right, all of them have a lot of mitochondria, and they require oxygen. Where does that oxygen come from? It comes from these little arteries that carry oxygen here. Where does it, they also need nutrients like glucose or fatty acids, fats, right? Where does that come from? It comes from the arteries that bring all of this stuff to the muscle cells, right? And so that muscle cell will take the oxygen, the fuel, and the little mitochondria in their cell will undergo what we call cellular respiration. They'll make a bunch of ATP, which is the whole point of this. They'll create heat at the same time, which is why we get warm when we use our muscles a lot. It'll also make CO2 and water. This CO2 needs to get gotten rid of somehow, right? It's a waste. So that'll go back in the veins actually, and they'll return back to the heart to be exhaled out. So we need a blood system to transport good things like oxygen and nutrients and to remove waste products, right? And we also need nerves to come to our muscles to tell them when to contract, okay? So that's the general idea here. Now I want to talk about the organization of skeletal muscles. Skeletal muscle has different connective tissue sheets around it. They call them sheaths, right? There's these thick layers around. So it's not just in a muscle organ, like a skeletal muscle, like a biceps muscle over here, right? You will see that there is the muscle fibers, right? There is muscle tissue, but there's connective tissue in between, right? Remember, organs are made up of many types of tissue. So over here, if I look at this muscle, right? This is the muscle in the arm over here. You see that, you have the tendon over here connecting it to a bone, and in this muscle, if we go in, it's a bit trippy, but there are like these cylinders inside of cylinders, right? And you see that this is the breakdown of what is the microscopic level of muscles. So this is like your big muscle that's in your arm here. You could probably plop that on the table, but these things over here, these smaller cylinders inside are much smaller. So I want to start with the lowest one. This is one muscle cell. This you would need a microscope to look at, right? But this is one muscle cell called a muscle fiber, right? Over here, this is one cross section, as if I chopped here, one muscle fiber. And you see that around it, there is this connective tissue. This is the actual, over here inside, you have the actual muscle fiber. You see organelles, right? You see some mitochondria inside. You see all of these myofibrils are organelles that are inside the cell. This is one cell. You see the nuclei over here. So this is one muscle cell. This is one muscle cell, one muscle fiber. And around it, you see a tissue, a connective tissue. It's kind of like an epithelium. There's always some connective tissue at the bottom. Well, here, the connective tissue is surrounding this, right? And so what do we call that? This is a delicate connective tissue sheath that's called the endomysium. Mesium means muscle. Endo means inside. It's the innermost connective tissue, okay? So you would see around this muscle cell is some endomysium here that's wrapping around and kind of cushioning around this one muscle cell. So there's an endomysium that wraps around an individual muscle cell. This is also called a muscle fiber, right? So this muscle cell has some endomysium around it. That's the first connective tissue sheath. Then here is one muscle cell, okay? And it is bundled together into a few muscle cells. Do you see this one, 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 this one? Here, if I chop this, right, I can count one muscle cell, 2, three, four, five, six, seven, 8 muscle cells, right? I see 8 muscle cells bundled into this grouping. I want you to know that muscle cells, they bundle together into groupings that are called fascicles. This is one fascicle. This is another fascicle, another one, right? So this is one whole fascicle that has many fibers in it, muscle fibers, muscle cells. So several muscle fibers are then wrapped together by a coarser, harder fibrous membrane called the perimysium. Peri is always the middle layer. So perimysium, the middle connective tissue layer over here, this is this connective tissue here, right? And this is one fascicle. One fascicle has many muscle cells inside, right? This one, you see its nucleus, and it's a bunch of its myofibrils. Its nucleus, many myofibrils, nucleus, many myofibrils. So this one thing is like one of these. Actually, this one thing, let's say it's this one here, right? So what's interesting, right, is remember I told you, what is it that's around one muscle cell? That's called endomysium. So all of this pink stuff here, right? Do you see all this pink that wraps around one muscle fiber? That's the endomysium, right? But wrapping around a bunch of fibers, here's a bunch of fibers, is another white layer over here of connective tissue. That's what we're going to call the perimysium. That's grouping together a few muscle fibers, right? All right. So this is like your little home, right? This is like the grouping of people on your floor. You have many people that might live on in apartments on your floor, right? But they each have their own house. That's a grouping. That's your floor. And then this over here is like as if there's many floors to your building, right? So there are actually many muscle fascicles, right? Many muscle fascicles. You see over here the endomysium inside between all of these muscle cells, right? You see the perimysium that is around all of the muscle fascicles, right? You see that it's going around the muscle fascicles. And then you see the last layer. So this is one fascicle, right? Here you have one fascicle. This is one fascicle. This is another fascicle. This is another one. And then lastly, several fascicles, right? All of these fascicles are wrapped by a third sheath, right? Here's the next sheath that surrounds all of these fascicles. And this is called the epimysium. Remember, epi means on top, the top layer. Mysium, the muscle, so the top layer of connective tissue on the muscle, epimysium. And this forms the skeletal muscle organ. This will wrap around and create whatever this muscle is, right? Let's say it's your biceps or your triceps, right? That'll form the outer layer. So here's your epimysium, making that outer layer of your muscle organ. That forms the skeletal muscle organ, right? So the epimysia then blend and form a strong cord-like tendon at the end. Once you get to the end of the muscle here, this epimysium actually becomes a different type of connective tissue that's tough at the end, and we call that a tendon. And a tendon kind of pulls and anchors down the muscle to the bone, okay? So we're going to do a little activity here. I want you to name what a muscle cell is, bundle of cells, and the organ. And I want you to tell me what connective tissue is directly surrounding it. Okay, so you can pause the video and take the time to fill this out. Okay, so over here, right, a muscle cell, another name we used for this was a muscle fiber, right? A muscle fiber. This is my one muscle fiber. This is my one muscle fiber. Then a bundle of muscle fibers, of muscle cells, right? If I have one muscle cell, two, three, four, right? What does that form? That forms our muscle fascicle, right? That forms our muscle fascicle. I don't know if I'm spelling that right. Fascicle. Yeah. Wow. I'm impressed. So this is my one muscle fascicle over here. I should write muscle, because there's nerve fascicles later on. And then if I take a bunch of muscle fascicles and bring them together, they'll form this one big organ, right? What's that organ? It's a muscle organ, right? That's why when you use the term muscle, you need to specify whether you're talking about a muscle cell, a muscle tissue, or a muscle organ, right? It could, or the muscular system, right? Remember the hierarchy levels, they change. So muscle organ, I'll give you an example, the biceps muscle, right? Or the triceps muscle. That's a muscle organ we often talk about. And now I want us to think about the connective tissue surrounding it. So the muscle fiber, if we look back over here, the one that's directly surrounding it, we can actually just go, we could go over here even. Here's one muscle cell, right? Surrounding all this is this pink stuff. That's the endomysium. Endo means innermost, right? Inside. It's the connective tissue layer that's the most inside, endo, inside, right? So we're going to call this the endomysium. Endomysium. Around a bunch of fascicles, right? So here's one fascicle, two, three, four. All around those fascicles, grouping together, is a connective tissue called the perimysium. Peri always means like the middle-ish layer, so perimysium. And I didn't tell you this, but look at the perimysium. It has a bunch of these little blood vessels and nerves going in it. That's good because they get to go to these muscle fascicles and start talking to them or sending them a bunch of oxygen from there. And then lastly, the muscle organ, what connective tissue structure surrounds it? Here's my whole muscle organ, and around it is a connective tissue. And it's epi, it's on top, right? Remember, epi means on the top layer, and epimysium on top of the muscle. So that's our outermost layer, epimysium. Epimysium. And as you get actually closer to the end of the muscle, however, it turns into like a tendon at the end. It's firmer. It anchors down. You don't need to know this for now, but a tendon, right? What is its use? It actually prevents the muscle from overextending and overstretching. It's kind of like a protective barrier so that you don't overexert your muscle. So this tendon doesn't contract. It kind of just anchors down and says, whoa, don't pull too much. So that's all we have for here. Now, some features of skeletal muscle fibers, right? So if we look at this one muscle fiber here, right? Only this one, or this one muscle fiber here, right? This is what we're going to see. A muscle fiber is also called a muscle cell, right? This is one cell. I know most cells we draw look like this, with a little nucleus, some Golgi over here, right? A little mitochondria, right? But this looks different, right? This looks like a long cylinder, and that's okay. We know that cells look a bit different depending on what type they are. This is a long and cylindrical shape. This is one muscle fiber here, okay? Do you see this one long cylinder? One muscle fiber here, right? This is a real microscope picture, by the way. One muscle fiber is multinucleated. It has many nuclei, right? Look here, 1, 2, 3 nuclei here. 1, 2, 3, 4, 5, 6, 7, 8, about here. So why do muscle cells have many nuclei? They actually need to make a lot of protein, so they need all that genetic information to constantly be giving the instructions to make the proteins, right? So the mRNA, it's constantly transcribing, translating so that it meets the high protein demands. It needs to keep growing and making some proteins inside to grow and to keep contracting. So we're going to see that that's important. We want, that's why there's a lot of nuclei. It's to meet the high protein demand. And what are those proteins that are high in demand, right? So a muscle fiber contains many small myofibril organelles, which are mostly protein. So these are inside one muscle cell, right? Is Are these myofibrils. You also see the nuclei, you see the little mitochondria, but there's also these humongous protein myofibrils here, right? So these are organelles inside the cytoplasm. This is like the cytoplasm of my cell, right? I know it looks weird. It looks like a cylinder all of a sudden. Like we think of cells usually and they're round and they're spherical, but this is a cylinder type of cell, right? And the myofibrils, you see this myofibril here, they're actually aligned. And they're aligned such that there's these dark bands and these light bands and dark bands and light bands, and they align in a certain way. Do you see all the darker bands aligning here? And then you see a light band align over here. And then it's a dark band and a light band. That's what creates the striations. The dark bands over here, we call them the A band, and the light I band, the light band we call the I band, right? So this is the I band over here, and then you have an A band, an I band, an A band, I, A, and on and on and on. So, how is an individual skeletal muscle fiber organized? This over here, right? This is as if I like sliced this in half and took out a little chunk here. This is a piece of this, right? I want us to know a few terms. So the plasma membrane, of course, we can't just keep the term plasma membrane. Biology needs to be annoying. For muscles, the plasma membrane of a muscle fiber is actually called the sarcolemma. We're going to see that the word sarco is very important for muscles. We'll see it again. And sarcolemma, a lemma is like the lining around. So it's the muscles lining. This is the plasma membrane. So this is the sarcolemma. You know, you could imagine just above here, all of this endomysium would be around this, and then you'd have this sarcolemma. I actually want to go back here. Do you see over here there's all this endomysium around, and then over here it says sarcolemma. This is the border around the cell, right? The little border that's around, and all around that border is some endomysium. So that's what we see over here. So this is my sarcolemma that surrounds. If I look here, I would call this my sarcolemma, right? It's another name for plasma membrane of a muscle cell, right? As this, and basically, there's a few other interesting things over here. Remember I told you that usually a nervous cell, a neuron, comes to the muscle and tells it to contract, right? So a neuron will often come down here and release some stuff to allow for contraction to occur. And as it releases this stuff, right, the neuron only comes at this level, let's say, but it needs to talk to these deeper little muscle cells to tell it that a neuron has told it to contract, right? A neuron only comes down to here, and it needs to tell these little myofibrils deeper inside that a neuron has told it to contract. Look over here. A neuron will just signal to these maybe over here, that it should contract, right? But look at these deeper ones here. How is that nervous signal going to get all the way down? Neurons do not enter into the cells here, right? So the neuron signal needs to somehow get deep here, and the way that this is done is through the spread across the sarcolemma. So This nervous signal will spread across the sarcolemma, right? The electrical signal will be propagated across here, and it will enter into what we call T tubules. Do you see over here these little pores, these little holes, right? These are the T tubules. So the electrical signals enter these little holes, bzz, bzz, and they get carried down these areas, and they could then trigger the inner myofibrils over here, over here, over here to contract as well, right? Because if a neuron just came here and talked to these myofibrils, only these would contract and the other ones would stay put. It would actually spasm your muscle. So you want all of the muscle, the whole cylinder to contract together, and that's why these T tubules these are important. They traverse or transverse across the muscle fiber, right? They go through deeper into the muscle fiber, and they can carry that electrical signal and distribute it to the depth of the muscle fibers, okay? The muscle cells cytoplasm, right, is called the sarcoplasm. I don't think we see it here, but I want you to look at this image. Do you see all of this? Here I said that there was the sarcolemma, which was the border of the cell, the plasma membrane, but all this yellow stuff in between the myofibrils is called sarcoplasm. It sounds like cytoplasm. And do you remember what cytoplasm was? Cytoplasm was the soup of the cell, right? Remember that, in our cell, in our cells, we saw in the previous lectures that there's a nucleus that's here, there's a little ER here, there's a little mitochondrion here, but all of this is floating in this stuff, this soup that's all around the cell, right? So the soup of this muscle cell is called not cytoplasm, it's called sarcoplasm. Remember I told you sarco has stuff to do with muscles, right? So sarcoplasm is a fancy word to say the cytoplasm of the muscle cell. If you look here, all of this yellow soup in between the myofibrils, that's what we call the sarcoplasm. So I want you to imagine a bunch of liquid, soup, jelly in between all of these organelles, right? Soup, you really, they're tightly packed, right? But all this stuff here, this yellow, and in between here, this stuff here, right? All of this is cytoplasm of the muscle, so we call it sarcoplasm. And as I said, there's barely, you don't see much of the sarcoplasm here because it's packed with contractile myofibrils. All of these myofibrils here, 1, 2, 3, 4, 5, 6, 7, 8, 9, they're packed in there, right? There's barely any space. This soup is full of noodles and not much broth, right? So it's filled with these myofibrils, and these myofibrils, they're organelles that are the contractile units. They actually are the ones that are going in and out, right? And since they're all packed here, they actually end up pushing nuclei to the periphery. That's why when you see these muscle cells, you see these nuclei here, these usually big nuclei, they're pushed to the sides, right? Do you see how here they're on the sides? Because they actually don't fit in the middle. These organelles push them so that they only go to the side. While the mitochondria occur between the myofibrils, the mitochondria are much smaller and they zigzag in between the myofibrils, but they're squeezed, right? So that's some terminology here. So we see our one muscle cell with a sarcolemma, a plasma membrane. It also has sarcoplasm inside, which is cytoplasm. There are T tubules for the nervous signal to go deep down into the muscle fiber. You see mitochondrion, of course, because these cells will need oxygen and glucose. You see, and the most important here is all the myofibrils, right? Not muscle fibers. Inside one muscle fiber are many myofibrils, right? Look over here. Here's one muscle fiber, and inside are many myofibrils, right? It says it here, myofibrils. There's many myofibrils. Those are organelles that contract, right? This is the thing that contracts. So, how is an individual skeletal muscle fiber organized? Each myofibril, right? Here's my one muscle cell, right? My muscle fiber. Here's the sarcolemma over here. Here is one myofibril, 2, 3, 4, 5 myofibrils, right? 1, 2, 3, 4, 5 myofibrils, right? I'm zooming in on that. Each myofibril is surrounded by a modified smooth ER. Remember, cells have nuclei, we saw those. They have mitochondria, we saw those. And in muscle cells, right, you have smooth ER, right, as we had seen. And it's called, but in this case, it's modified a bit. In muscle cells, I want you to remember what smooth ER was for. We saw that one of the functions of smooth ER was to produce some lipids, right? So to manufacture certain types of lipids. I recall us saying at one point that smooth ER is also very useful for storing different ions like calcium. Muscle cells rely on calcium like crazy, okay? So muscle cells have a crazy amount of smooth ER, like much more than any other cell, right? When we talked about smooth ER in a regular cell, we kind of just brushed over it, but these calcium containers, these are very useful in skeletal muscle, and we're going to see that in skeletal muscle, there's actually a modified smooth ER, so that's huge, it's bigger, and we call it, because it's modified, we call it sarcoplasmic reticulum. We don't call it endoplasmic reticulum, we call it the sarcoplasmic reticulum. It's special smooth muscle ER just for the muscle cell. It's the same thing, right? It's just that we give it a name because it's special in muscle cells. So you see that over here, right? You have these, so you have your different myofibrils, right? 1, 2, 3, 4, 5. You have your sarcoplasmic reticulum that wraps around it, right? We call it the SR sometimes. So the SR actually connects to the T tubules. Do you remember the T tubules? These yellow T tubules here that go deep down. Here you see them go down and they actually meet over here. Do you see the sarcoplasmic reticulum now? Right? We added that piece to our puzzle now. You see them close to the sarcoplasmic reticulum. Where the SR contacts the T tubule here, right? This is the T tubule that came in, you see from the plasma membrane, it comes in and it goes deep down, right? Here's my T tubule. Where the SR contacts the T tubule, which is over here, or all of this here, right? Do you see the dark blue is the SR? We call the SR at that region a terminal cisterne. Camion cisterne in French is a truck that holds a bunch of stuff, liquid usually. So like a water truck, right? That has a big bin in the back for water. That's a camion cisterne. So this is like a big area where we can hold a lot of calcium in this case, right? So terminal because it's at the ends of the smooth ER, and cisterne because it's like a camion cisterne. It's swollen and it holds a bunch of this calcium, right? It's like it's a not a water reserve, but it's a calcium reserve in this case, right? And it swells up at the ends here, right next to the T tubules, right? And actually, two terminal cisternae, one on this side and one on this side, sandwiching one T tubule equals a triad. We call this a triad. The triad is the one T tubule in the middle and two terminal cisternae of the SR, sarcoplasmic reticulum, on each side. So that's a lot of stupid vocabulary, but we're going to get used to using them Just make sure that you understand the vocabulary before moving on. Okay, so here's my triad on this side, right? Um, and I'm going to put this into context. Remember what the T tubules are for. Over here, when a neuron sends a signal to the muscle cell, I told you that that signal travels down deep into the muscle, right? So that signal might travel down over here, and it might tell the cisternae that are filled with calcium to release it, right? I'm giving you a sneak peek of what's happening. When the signal on the outside from the neuron or the nerve cell talks to the muscle, right? When you tell your muscle to contract, right? Well, that neuron sends a signal down to your muscle, and it will go down deep into the muscle and tell these areas to release calcium that will cause the muscle to contract. We'll see that in a bit. So that's a bit of terminology about the ER that's here. So we talked about how the muscle fiber was organized, how this big guy over here was organized. Now I want to delve deeper. I want to look at how the myofibril is organized. How is a myofibril organized? Here is my one muscle cell. Here is one myofibril. And now I want to zoom in onto just this area of the myofibril. Okay, I want to zoom in deep, deep, deep. One myofibril is made of two types of myofilaments. Okay, so in a myofibril, one myofibril, I know the names are not, I didn't make these names up. In one myofibril, you have many myofilaments. Fibril sounds bigger. Fiber sounds big. A big fiber. A fibril sounds smaller, and a filament sounds super small, right? So we have the fiber, which is the cell. We have many myofibrils, and in one myofibril, we have many myofilaments. So if I zoom into one myofibril, I will see, do you see all these over here on the side, the cross section? You see all these small little dots? These are all little myofilaments, little fil in French, right? And there's actually two types. You see blue ones and you see red ones here. The blue ones are called actin. They're not actually blue, they're just colored this way in this design. So these are the thin filaments. A study tip is actin is thin, very thin, okay? I really like it. So this thin filament of actin, right, is, you see it's thinner and it's on the outsides here, right? Let's just zoom in onto like one thing like this. You see the actin is on over here, it's thinner. And then you see this myosin is the thicker filament, right? Myosin is this big, thicker filament. You see these big red ones in the middle. They don't really go bigger or smaller. We'll see that in a minute. So you have actin and you have myosin here. Okay, so that's all I need you to know for now. We have a muscle fiber, we have myofibrils, and inside the myofibrils are myofilaments. And those myofilaments, some of them are called actin, and some of them are called myosin. Actin is thinner, myosin is thicker. That's all for now. I know it's a lot. I'm not downplaying it. That's what I said. It's not complicated, the muscle system, but you need to organize your head into how it gets smaller and smaller and all these myo names. Now, what is a sarcomere? A sarcomere is the smallest contractile unit of a muscle. We call that the sarcomere. And so the sarcomere is repeated along the entire length of the myofibril. Here's one myofibril. This here is one sarcomere. It's from this Z line to this other Z line. And this is the M line. This is like the middle line. But we're going to see that the actin, they connect over here at the ends, right? They don't touch each other. Do you see how this blue actin doesn't touch the other side? That's because these actin will go in and out, and they never touch each other, okay? But these blue lines will actually come in closer. We're going to see that in a minute. But I just want you to know for now that the sarcomere, this is the smallest region that we can say contracts and relaxes. The whole muscle contracts and relaxes, but this is the smallest unit that contracts and relaxes. It's like a boat, right? In a canoe, right? You're a bunch of people in the canoe. The whole boat is moving, right? But each of the individual people are the smallest unit that are all paddling, right? So the people paddling are these sarcomeres, right? This is one paddler, the sarcomere here. This is another paddler. Do you see this grouping here? You're going to have another paddler here, and you're going to have another one here, right? Each of these groupings, right, from this blue line to this blue line, this is one person in the canoe, another person, right? So that's the smallest unit that paddles and contracts the muscle. All right, so the sarcomere. Do you see sarco again? That means muscle. That's where the sarco came from. The sarcomere, it's the smallest unit in the myofibril that contracts. So, before we go on, I want you to pause this video. I want you to make sure you know the organization of these things because it gets messy. Arrange the following from the most superficial, meaning superficial means out there, up and fake and well, that's the personality definition, but superficial means at the top, right? You think you're the top of the world. From the most superficial to the deepest, deep inside, right? So we want to start from the outermost down into the innermost. You want to put the right order of these. So from superficial to deep, you're going to put the layers, right? And then here you have fascicle. Then you have two more, and then myofibrils, and then something else, and then the deepest layer, right? So I want you to pause the video and try to do this on your own first. All right, so I don't see any muscle name like the biceps or triceps, right? So I'm going to start with the outermost connective tissue. That's the epimysium. So I'm going to put B here. The epimysium is the outermost layer, right? That defines the boundaries of like the biceps muscle. Then, so all that epimysium, right? Then, let me go back over here. You have this big, let me erase here. You have the epimysium, right? And it holds all of these together, right? Then you have the fascicles. Around each of the fascicles are the perimysium. So I'm actually going to go over here and write the next one is the perimysium, C, right? And that groups fascicles into bundles, right? It actually surrounds each of the fascicles. And from the fascicles, right, if I go back to my little sheet here, you see this is like one fascicle, right? And in one fascicle, all of this connective tissue in between, right? Remember, all this connective tissue in between is called the endomysium, right? So I'm going to go and put after the fascicles, right? You have this endomysium, that's D. And then, right, now that we have this endomysium that's wrapping, let me show you right over here. You have all of this endomysium wrapping around, right? Remember the next layer, as we're going inwards, right? We went through the epimysium. We went through the perimysium. We went through the endomysium. Now, so through the endomysium, and now we're going to enter the actual plasma membrane of the muscle, right? What is this plasma membrane called? Remember, it's called the sarcolemma, right? So the next thing that we are going to get through is E, the sarcolemma. And then, right, we're going to reach the myofibrils. Actually, you could add from the sarcolemma, you enter the sarcoplasm, the cytoplasm of the muscle cell. And then you're going to enter into these little myofibrils, right? So The next thing here is myofibrils. You enter into the myofibrils. Here's the myofibril, right? So you enter into the myofibril, and then you will touch a bunch of these little myofilaments. So this is where we would have myofilaments A over here. You'd have these myofilaments. And then you're in the deepest layer, right? Perfect. This helps to organize things in our head a bit. So that was how the organization of this, how the skeletal muscle is organized. This is key before you move on to the next parts, because understanding that vocabulary and where things are and what a myofiber is, or a myofibril or a myofilament, it's going to get messy. So I want you to go back to this part 2 if you need to review it, right? Make sure you understand the vocabulary, and then we apply our knowledge, our physiology over here in the next few parts. So the first thing I'll talk about is the sliding filament theory and the sarcomere. So, how do we characterize the sarcomere? Remember I told you that a sarcomere is from one Z line to another Z line, right? So this is a sarcomere is the arrangement of actin and myosin filaments that create striations seen in skeletal muscle. Do you see these darker regions, these lighter regions, darker, lighter? The zebra stripes, right? Do you see over here the zebra stripes? They come from the arrangement of the actin and the myosin. So here, in these lighter regions, you have the thinner fibers, right? And in these darker regions, you have the thicker fibers, right? Remember I told you that the actin is thin, the myosin is thicker, right? So these striations, right, they're composed of different bands, okay? So let's go through the bands over here. Let's look at one sarcomere. One sarcomere is from this end to this end. That's one sarcomere, okay? So the sarcomere is from really from here all the way to here, okay? That defines one. You should know what one sarcomere looks like. In one sarcomere, we have the I band, okay? So this is the light band, the area with just actin. Over here, do you see just the actin, right? Do you see over here? Forget about the yellow stuff. That's just an elasticy fiber. Here in this area, you see just the blue actin fibers. Do you see how the actin fibers, they're anchored over here to this Z line here, to this end over here? And these actin fibers, they are anchored over here to the end. The myosin fibers, the thick fibers, they're in the middle and they're anchored to the M line here. I want you to know that the myosin fibers don't actually move in terms of position. They stay put. The little heads on top here will move. We'll see they kind of row, but it's really the actin fibers. It's as if here is my myosin. Here's my myosin fiber. The actin fibers, they move on top of the myosin one. They pull inwards and outwards. The myosin fibers are going to actually be the ones that pull the actin fibers in and out. So I like to think of actin as kind of like my two hands here, and they pull out, they pull in, they pull out, they pull in, right? So think of here as one set, one arm of the actin fibers. Here's another arm of the actin fibers, and they go in and out, right? So this whole unit, this whole sarcomere, right? One sarcomere, this side gets pulled in this way, and this side gets pulled in that way, okay? Now, I just want us to go through the anatomy, the bands, and where everything is. So we call the I band the light band. And if you look at the light band here, this is the lighter region, right? This is just thin filaments. Actin is the thin filament, right? So all of this here is the I band. All of this is the I band. Forget the yellow again. That's just a little elasticy protein, but we don't see it. So you see all of this actin, actin filaments. The H band is the area with just myosin, just the thick filaments. Okay, so here, this is the H band here in the the middle here, this is where there's just the, this is where there's only the thick myosin filaments. Then we have the zone of overlap. All of this here is a zone of overlap because you have actin and myosin, actin and myosin, actin, myosin. And then we have what we call the A band, is the dark band, the true dark band, is an area including the entire myosin filament. So the A band goes from where the myosin is to the other end where the myosin is. It includes the H band and the two zones of overlap on each side. Okay, so here's the deal. What defines the middle and the ends of the sarcomere? The sarcomere's two ends are called the Z lines. Here, this is the Z line, and here, this is the Z line here, right? It is the point of attachment for actin filaments. Do you see the blue actin filaments attaching to this Z line? Here, these blue actin filaments attach to this Z line. Here you see the blue actin filaments attached to this Z line. Here the blue actin filaments attached to this Z line, right? So the actin filaments, they attach there so they're the attachment point for actin filaments, and also for titin. This yellow protein here is called titin, and it attaches to the Z line as well. We'll talk about in 1 minute what the titin does, but I just want you to think Z line is, Z is the last letter in the alphabet. So these are the ends of the sarcomere, right? This is one end of the sarcomere, the other end, Z is the last letter of the alphabet. It makes sense. So you have two Z ends over here. The elastic filament, this yellow thing, is called titin, and it actually anchors the myosin to this, to the Z line. The myosin does not contract or move, right? It actually stays put there, and it's held there with the titin, right? It's holding it tight. So it's just an elasticy kind of bungee cord that holds this straight and up. The actin is directly touching the Z line, so it's good. The only other thing to add here is the M line. This defines the middle of the sarcomere. M is for middle, thank goodness. So this is the M line over here. This is the middle, right? This middle section. It's the point of attachment that anchors the myosin filaments to the middle, right? So these myosin are anchored here, and they're pulled on each side by this titin, but they don't move. Like, they don't stretch more than needed. They stay as is put. It's really the actin that's going to move on top of it. Okay, so how does the sarcomere change during a contraction? Okay, so this is a fully relaxed sarcomere of a muscle fiber. I want you to imagine your biceps muscle, right? And it's relaxed. And then I want you to imagine it when it contracts, right? When it's relaxed, right? All of those muscle fibers have myofilaments, which have sarcomeres, right? And each of the sarcomeres, when it's relaxed, look like this, okay? And then when they're contracted, this is what the sarcomeres are doing, okay? So here's the deal. Myosin is anchored to the center of the sarcomere over here. Do you see these big, thick filaments? It's centered over here at the M line. And it moves their heads to pull on the actin filaments. Do you see these myosin heads that are on these thick myosin filaments? A study tip, myosin sounds like the medusa, right? A medusa has many headed little snakes on her head. It kind of looks like this, right? So many little heads. And these little heads, I want you to think of them like oars that, or little hands that are grabbing onto the actin. They're actually going to grab onto this actin here and pull it, okay? So these heads grab onto the actin and pull it. Actin is anchored to the center of the ends of the sarcomere. So actin over here, right, this blue is anchored to this Z line here. And myosin will pull it inwards towards the M line. So here's my myosin heads. The myosin will always pull it into the middle to the M line. So these heads here, they grab onto the actin here, and they will pull it this way. These heads here grab and pull. These heads here grab and pull. These heads here grab and pull, and they pull the Z line inwards. These heads here are pivoting the different direction. They're taking this and pulling it inwards. Taking this and pulling it inwards. Taking this and pulling it inwards. So all that the myosin does is the little heads, they move back and forth like this, and they grab onto the actin and pull them in, detach, remove, pull, and on and on. Whereas the actin over here, right, the myosin will pull it. So here's actin, right? These are little actin subunits that are on, these are the filaments, right? And you have little myosin binding sites on these little blueberries. And the heads of this myosin bind onto the areas over here of the actin to pull it in. So I want you to look at this picture very well. Here, when we're relaxed, look at how big this fiber is. Look at the Z line over here and over here, right? Do you notice that when we contract the muscle, right, these Z lines come inwards, and look at where the Z lines are now. They're closer in. Now I want you to take 2 seconds to compare the myosin. Look at where the start of the myosin is here, and look at where the start of the myosin is here. Look at where the end of the myosin is here and where it is. Did it move? No. These little heads went back and forth, right? But they didn't move. The M line neither didn't move. What is it that moved? The Z lines moved inwards. Why did the Z lines move inwards? Because these little heads grabbed onto this blue actin and pulled it inwards, right? Do you notice how the blue actin filament is now over here? It's touching in the middle, right? It got brought, I like to show it like this, it got brought inwards like this. It started kind of on the outskirts like this, and it went inwards, and it brought the ends of the walls inside, right? So this is relaxed versus contracted, right? This is what happens when your muscle contracts. So I want you to pause the video here. I want you to draw arrows to label at least 2 myosin filaments, and draw arrows to label at least 2 actin filaments. And then when the muscle contracts, which filament gets closer to the center of the sarcomere? Okay. So you can pause the video and take a crack at this. So over here, draw arrows to label at least 2 myosin filaments. Over here I see this over here is 1 myosin filament, right? And then I'll go and do this one too. This is a second myosin filament too. And this is part of this one sarcomere here, right? I could go to this sarcomere over here and draw it too. Here I see one over here, I see another one over here, right? To another one here, another one here. So those are some myosin filaments. And then I want us to draw arrows to label at least 2 actin filaments. I see one here, and I see another one here, right? I could choose these 2, one on this side, another one on that side, right? Oops. So I see one on this side, one actin, and another actin on this side. When muscles contracts, which filaments get closer to the center of the sarcomere? That's going to be the yellow actin that will get closer into the middle, right? The purple, the myosin, will not move, but the yellow actin will move inwards. So actin is the answer here. This yellow one will get closer in, and these lines here will get brought inwards, whereas the purple here will stay put. And the little heads here will row inwards. All right, so over here, Now let's piece it all together. I think I was struggling to bring this all together. I think now is going to be a good spot to do it. How does the sarcomere, right? Remember what a sarcomere is from Z to Z, right? How does that contract? You see that here from Z to Z, it's this distance. Then all of a sudden from Z to Z is a much smaller distance. It got contracted. How does that happen? Because here you see a fully relaxed sarcomere, and then this is fully contracted. So during a muscle contraction, the Z lines are pulled closer towards the M line. These lines here on each end, they are pulled towards the middle, right? Here are my Z lines. They go in towards the middle, right? Here are my Z lines, here. They become closer to the middle. The middle doesn't move. The H band shrinks because the actin filaments are pulled closer towards the M line. What is the H band again? The H band is just the line, the band that has only myosin. So the band with only myosin actually gets smaller because look, over here, you see that this is where the actin was, right? And then afterwards, the actin moved inwards like this and covered up the only myosin area, right? So here you see that it's covering now this area that used to be free, right? Now the actin is actually covering it up. So the H band shrinks and actually disappears when the actin fully comes to the middle because they're being pushed in towards the midline. And the zone of overlap gets larger, right? Here is my zone of overlap. This, let me put it in a different color. This is my zone of overlap before, right? But because this area, the actin is going to move on to this area, this is now my zone of overlap, right? So this was my zone of overlap before. Now all of this here is my zone of overlap. Okay, so the A band remains the same size as it includes both the zone of overlap and the H band. Remember, the A band has this zone of overlap plus the H. So remember that the myosin does not change in size, so it always stays as is, right? It's just that the zone of overlap gets bigger and the H band gets smaller, so it's relative. And lastly, the I band shrinks because the actin filaments overlap more with the myosin. This used to be my I band here, right? Where I have just actin, but now my actin is moving onto this myosin and it's creating this much smaller I band. So that's because the actin overlaps more with the myosin and it is creating more of an overlap, the overlap band, right? The zone of overlap. So why do the bands and lines change the way they do? The simple answer is that the myosin filaments, which stay put in the middle, they pull on the actin filaments, these blue filaments. These heads pull on the actin filaments on both sides. So these ones pull on these actin filaments and they push them in to the right. And these myosins, right, they hook on to these blue actin and pull them in towards the left. And this will effectively shrink the entire sarcomere, right? I want you to think of this as a tug of war, and they're pulling, these these myosins are pulling inwards, and these myosin filaments are pulling these inwards, right? And both the Z lines will shrink. So I want you to just look at the animation right now, right? This is what happens. The Z line, right? Look at each of the Z lines, right? The myosins, you don't see them pulling right now, but they do pull, right? Both of them inwards, effectively shrinking the entire sarcomere, right? Now my Z line is here, and so my sarcomere is shorter. So I've, when you look at your muscle and you contract it, that bicep muscle that bulges out, right? That's because all of the sarcomeres have come inwards and kind of accumulated there in that middle area. That's what a muscle contraction is. It's this happening many times. Because remember, this is just one sarcomere. There's many sarcomeres in one myofibril, in one myofilament, and there's many myofilaments. So all of them going together in unison. Okay, now, over here, I want you to pause the video, and this is actually a real electron microscope showing the filaments, right? And so I want you to mark here the various things that they ask. A band, I band, H zone, the M line, Z line, the actin, right? And then over here it says use the table to describe each component change in size during a muscle contraction. It could get bigger in size, smaller in size, or not change in size, okay? So pause the video and take a crack at this. All right, so what we're going to do, the first thing that they want us to label is the A band, right? And so if we remember correctly, the A band, right, is, let's go back here. The A band is the zone of overlap plus the H band. So it's the whole region where the myosin is, right? And remember, myosin is the thicker, darker filament, right? So all of this here, right, I'm going to label all of this as my A band. Remember, the I band is the light band. And the I band, you could say for this sarcomere, right, it's over here, but it also, for the other sarcomere, it's on the other side. So this is the I band. If you were just talking about this one sarcomere, though, this would be our I band, or our only our thin filaments, our actin, right? So I band, and here's the I band of the next sarcomere on the other side. And then the other thing that we need to do is look at the H zone. So the H zone is this area here, H band, right? This is where you find only the myosin. So this over here would be the zone of overlap up until the H zone. Mark the Z lines. So I'm going to go here and mark the Z disc, they call it here, the Z line. That's the ends over here of each. And I'm going to go mark the M line, right? The M line, I'll do it in another color. The M line here, right, would be this invisible line in the middle, right? The very middle. And then draw a line to represent the length of one actin filament, right? One actin filament would go, so let's look at the actin here. That would start at the Z line, and it would go all the way in, right, up until here, not in the H band, right? This is the zone of overlap here. This is the I band. This is the H band where there's only myosin. So same on this side. I'm just going to go from here all the way to my other one. This is the length. This is one actin, right? And this is another actin on this side, right? They're separate filaments. They don't connect to each other, but they're on opposite ends. And then in the table, describe if and how each component changes in size during a muscle contraction. So let's go one by one. The A band, does it get bigger, smaller, or not move? The A band is the overlap plus the H band. As the overlap increases, the H band decreases. So actually the A band stays put. The A band is the region where myosin is, right? So for the A band, I would say no change during a contraction, right? No change. The I band, right over here, is only the actin thin filament. And as your Z line comes in, right, it gets smaller and smaller because there's more overlap. So the I band actually will decrease in size. The H zone here, which is only myosin, remember the actin comes over and starts taking over the H band and making it smaller and smaller, so that would decrease. The Z line, right? So for the Z line, there's no change. It will always be, this is the where the Z line is, right? You can't change that in your whole little system here. So this is no change. If I said the distance between the Z lines, right, then I would say that that would decrease because from this Z line to this one, that will decrease, but the Z line itself stays put and doesn't change. And then the M line always stays in the middle, right? So that has no change, okay? Good. Amazing. Sweet. So that was the sliding filament theory and the sarcomere. So now we understand at the microscopic level what's happening, how a muscle contracts, right? It's not as simple as we thought it was in the beginning. So now we're going to talk about the neuromuscular junction. I went from big picture muscle down, 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 down to the nitty gritty. Now I want us to go back out to the muscle, right? And think about how it gets stimulated. I'm telling you that these fibers go in and out, in and out. But how is it that they're stimulated to do that? Because muscles don't do their contraction on their own. They get stimulated to do that, right? So for that, we need to talk about the neuro, brain, muscular, muscle junction. How does the nervous system talk to the muscular system to tell it to contract? So what stimulates myofibrils to contract? What stimulates actin to move on top of the myosin, right? And to talk about that, we need to define the neuromuscular junction. It's the region where a motor neuron, a neuron that will go and give a motor command to the muscle, where that motor neuron reaches its target and contracts the skeletal muscle. These are muscle fibers. This is one muscle cell, two muscle cells. These are skeletal muscle because they're long, they're parallel, they're striated, right? Very interesting. And on top of it, do you see this? This is the motor neuron. This is a neuron that's coming down from the spinal cord, from the brain, and it's sending its projections all over these muscle fibers, right? So the neuromuscular junction is the region where the motor neuron reaches its target and contracts the skeletal muscle. It consists of multiple axon terminals. So this axon here is the branch of the neuron, and it splits into many terminals, right? This neuron that came up from the spinal cord, from the brain, it's coming down and then it splits. So there are many axon terminals of the nerve, right? So the neuromuscular junction consists of these terminals plus the sarcolemma of multiple skeletal muscles. So in black here, you see the terminal, one terminal of the nerve. And over here, you see all of this pink region around of the skeletal muscle. That's part of the neuromuscular junction. Over here, you see all of this black area. That's the motor neurons, another piece of the axon terminals that branched out. And you see all the pink around. That's the skeletal muscle, right? That's part of the neuromuscular junction for this neuron, for this nerve. Over here, you see this little terminal and all this pink region. That's part of the neuromuscular junction. It's the junction between the neuron, the nerve, and the muscle. And because it branches out like this, there are sometimes you could actually have many skeletal muscles that belong to one neuromuscular junction. All right, so now we're going to talk about what is the neuromuscular junction. Sometimes it's abbreviated as NMJ. The neuromuscular junction is the connection between a neuron and a muscle fiber. This is usually always a motor neuron that's going to the muscle to allow it to contract. It's sending a signal down. So over here you see the brain, right? Signals probably sent down to this neuron in the spinal cord, and you see this neuron go all the way to this biceps muscle, right? Remember, I told you our biceps muscle doesn't contract on its own. A signal from the brain down the spinal cord comes to tell it to contract. So here you see this, what we call the motor neuron, because it's going down to the muscle to tell it to contract. That's going down over here. And we call this the axon. This is carrying the signal down to the axon terminals over here. So over here, you see this is a zoom in on this area, and you see the axon of the motor neuron come down, and it branches into these little parts here, right? These are called the axon terminals. These are the ends of the axon, and at it, when you look and zoom in, you will find the neuromuscular junction. It is the junction between the neuron and the muscle where they meet, okay? So over here, if you zoom into this little piece over here, right, the electrical signal travels bzz, 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 all the way down. When you decide to move your muscle, you send a signal from your brain down the spinal cord. It'll go all the way down this axon to the muscle that you want to move, right? If you want to go grab a coffee, right, you need to contract your biceps muscle to reach it. So that signal starts here, I want coffee. It sends down that signal to your biceps to contract to reach out for the coffee. So this is going all the way to the end. And here you see the end, right? This is like the axon of the motor neuron, that wire that's coming down. And here is a terminal, the axon terminal. It's a blip, a blip in a little bulb of the axon, right? So they kind of bulb out like this and form these um, little bigger parts at the end of the terminals. So um, over here, right? Let me just go through something first. I want to talk about what is a synapse, because I'm going to use this word a lot, or sometimes we use the word synaptic cleft. So a synapse is a space, okay, or synaptic cleft. Cleft means a space, right? Maybe you've heard of cleft palate or cleft lip, that's split in two, right? That means split apart, right? So the synaptic cleft is actually a little space here. The neuromuscular junction, the junction between the yellow neuron here and the red muscle here, there's actually that junction is not directly physically connected. There is a space called the synapse. So here you see the motor neuron, this neuron coming down from the brain over here, right, the axon, and it ends at this synaptic terminal. Well, you see that this over here is the sarcolemma, right? The plasma membrane of the muscle, and it is not touching this yellow plasma membrane of the neuron, right? So there's actually a distance between this neuron over here and the plasma membrane, the sarcolemma over here. So this space is what we call the synapse, and so the synapse describes the space between the axon terminal and of the neuron and the muscle's sarcolemma, the muscle's plasma membrane, right? Also, inside the little axon terminal over here, you see these little dots, right? These are called neurotransmitters. They are little chemical messengers that are released by the neuron. So the neuron actually stores all these neurotransmitters here, and they'll release it from the neuron into this space here, into the synaptic space. So when a signal comes down from the brain and tells this neuron to release the neurotransmitters, right, to release the chemical messengers, these neurotransmitters will be put into this synaptic cleft here, okay? In our case, there are many types of neurotransmitters. You might have heard about dopamine, serotonin, all of these. Well, in our case, we care about acetylcholine. For skeletal muscle, the neurotransmitter that gets released onto skeletal muscle is called acetylcholine. So these green dots here, they're neurotransmitters. Specifically, they're called acetylcholine, sometimes abbreviated like this. They are going to be released when a signal tells from the brain says to contract a muscle, that electrical signal will travel down and tell these little vesicles, right, synaptic vesicles full of acetylcholine to release into the synaptic cleft, and that will then cause something in the muscle, okay? And that's where we talk about the action potential. Once these neurotransmitters are released, they bind onto these receptors here. Do you see these purple receptors? And they will cause what we call an action potential. Potential, maybe you've heard of the word potential difference in physics when you learned about electricity, the potential of something, right? That's the voltage of something. So it's an electrical potential energy. So an action potential is actually a wave of electrical signals. Once these acetylcholine bind to the receptors, they open up and they allow charges, actually, it's ions that like sodium to flow in, and these moving charges can then cause, this is why I put these little lightnings here, they cause an electrical event. When the charges move inside, they cause a change in the voltage here, which can cause what we call the action potential bzz, bzz, to then move along over here in the muscle, right? Technically, an action potential, an electrical, this electrical signal wave comes down the axon and reaches the terminals over here, and then you see these chemicals get released. The chemicals bind to the receptors and they cause an action potential on the other side, in the muscle side, right? So all this to say, the neuromuscular junction is the junction between the neuron and the muscle. Here we had electrical signals come down all the way down to the end. A chemical signal got put into the synaptic cleft, into the synapse, into the space, and that chemical bound to a receptor to open channels so that ions like sodium can flow in and cause another electrical signal, but on the other side of the junction, right, on the muscular side. And this wave of electricity is then going to go through through the muscle, okay? So I'm going to go through the steps briefly, and then we're going to go more in depth into it. Again, if all these words are making too much sense now, it's a bit normal because these are nervous system terminologies. We're going to see it much more in depth in the nervous system, but I want us to start getting used to what a synapse is, what a neurotransmitter is, an action potential being like a bzz, bzz, electrical wave. We'll talk about more detail what it is, but first I want us to start getting used to these terms. So the first step is that a motor neuron from probably up in the brain, spinal cord, fires an action potential down the entire length of the axon. So all this electrical wave comes down. That starts in the brain because these are voluntary muscles. You decide to move your arm, so it starts up here. Then step 2, the motor neuron's axon terminals, the ends over here, they release acetylcholine out into this small space that's called the synaptic cleft, right? So this is our neurotransmitter that's released into the space here. Acetylcholine then binds onto these receptors, these purple receptors, that are channels on the sarcolemma, on the plasma membrane of the muscle. And they will open up and cause sodium. You don't see that in this picture. Sodium will rush into these channels. Sodium is a plus charge, and that plus charge will cause an electrical change in the muscle, right? And that's called an action potential. So that will cause an action potential, which will then go down the muscle fiber and stimulate it to contract. Now I'm going to go through a bit more detail on the neuromuscular junction steps. From the stimulus being sent down the motor neuron all the way to that wave, that action potential wave, I want us to know a bit more detail, certain channels that open up, like what causes this electricity. So over here, step 1, an action potential from the motor neuron arrives at the axon terminal. So you see it, this is the axon of a motor neuron coming from the brain, and it arrives at these synaptic terminals, right? At the axon terminals of the neuromuscular junction. Then this will cause, let's zoom in on that. So here you see this little pink wave, the action potential, the electrical signal arrives down. This depolarization just means electrical stimulation, okay? This depolarization causes calcium channels, these purple calcium channels, to open up, and this causes calcium from the outside of the cell, where it's very high in concentration, to move into this region of the neuron where calcium is lower, right? So calcium moves into the terminal via diffusion. Um, Calcium entry causes vesicles containing neurotransmitters of acetylcholine to be released. So the way I like to think of this is that calcium, we know that calcium is important for bones, it's important for muscles. I think of it as the moving ion. When it comes in, it triggers a bunch of events that we don't need to know about, but it is kind of it helps with this moving of this vesicle, and you see them being fused over here with the end of the neuron, right? So calcium entry causes these vesicles to move to the membrane and exocytose into the synapse. So do you see all this calcium, the green that's released here? That's because calcium entered. It told the little vesicle full of acetylcholine to fuse, and now there's full of acetylcholine in the synapse. Acetylcholine then diffuses across the synaptic cleft, right? And it binds onto these acetylcholine receptors. Do you see these purple receptors? These are not the calcium receptors. These are acetylcholine because you see the green binding onto them, right? They're specific for acetylcholine. And these are located on the sarcolemma, right? On the plasma membrane of the muscle. Do you see here the sarcolemma? This is of the muscle fiber, right? So you see the acetylcholine binding onto this. Then, right, acetylcholine binding on this receptor will open these receptors, these channels. These channels are sodium channels. Remember, this was calcium channels? When these open up, they allow sodium to come in. You see that in this step here. Once this acetylcholine in green is bound onto these sodium channels, it'll open the passageway so that sodium can come in and cause this little electrical depolarization of the muscle fiber. It causes it to be a little bit more electrically positive on the inside because this is a plus charge, right? So that's called a depolarization. Step 6, right? This lasts until the acetylcholine is gone. So actually, there's acetylcholine's effects, they're terminated by its breakdown in the synaptic cleft. If you leave this acetylcholine over here in the synaptic cleft, this will actually cause the muscle to keep contracting, and this will cause a stiffening of the muscle, right? A muscular spasm. So actually, once you're done contracting the muscle, you need to stop doing that. The way that this is done is another little enzyme that's found on the sarcolemma, right? This is called acetylcholinesterase. So acetylcholine esterase. ACE means it's an enzyme. This is a chopping enzyme that will chop the acetylcholine. So this over here, this little enzyme, right? Do you see it taking the acetylcholine and chopping it, degrading the acetylcholine? This is useful because it'll stop the signal, right? And also, the way that acetylcholine stops doing its job is it'll bind and unbind, bind and unbind, and eventually it diffuses away, right? So it'll get lost and it won't be in the synapse anymore, so it'll stop. If there's not another signal that comes down to release more acetylcholine, then the muscle will stop. If there is a continuous neuron, like your brain keeps saying to hold onto this object as much as possible, that neuron will keep sending signals to contract the muscle, right? Imagine you're holding a heavy box and you don't want to let go of it. Your neuron will keep sending this signal and keep releasing acetylcholine. Even though it keeps getting broken down, it'll sustain the amount of acetylcholine. And then when you let go of the box, right? And you stop contracting, well, the little acetylcholinesterase here will break up all the acetylcholine and you will stop having acetylcholine in the synaptic cleft. Okay? So I'm gonna let us watch this video.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
All right, so in this question, it asks interfering with the function of acetylcholine at the NMJ will interfere with muscle function and can lead to death. 2 ways to interfere with acetylcholine function are to block acetylcholine receptors or to inhibit the enzyme acetylcholinesterase. Predict how each will affect muscle function. So I'll let you pause your video and try to take a crack at this. All right, so the effect of blocking the acetylcholine receptor, right? That would be this little pink receptor here where the acetylcholine could bind on. What effect will that have? Well, if this is blocked, right, then acetylcholine cannot bind onto this receptor, right? And then this receptor cannot open, sodium cannot go in, and therefore the action potential, the electrical events in the muscle cannot occur, and therefore contraction will not happen, right? That's the effect of blocking the acetylcholine receptor. What about the effect of inhibiting acetylcholinesterase? Acetylcholinesterase, you don't see it here, but it breaks down this acetylcholine, right, when it's in the cleft, and it is responsible for stopping the signal, right? So if we block acetylcholinesterase, the scissors that break down acetylcholine, then there will always be acetylcholine in the synapse, right? Meaning it will always be there, always be binding onto these channels, and therefore always signaling electrical events, right? So actually here, right, for the acetylcholine receptor, when we block these over here, right, this would cause an inability for muscle contract, right? And when we block the little scissors, you don't see them here, but when you block the scissors that break down acetylcholine, let's say that these receptors are now all okay and they're working well, right? But the little scissors that break down acetylcholine don't work, the acetylcholinesterase, well, you would be stuck with a bunch of acetylcholine in the synapse, right? Meaning that it would continuously bind, continuously send action potentials, electrical events. So actually, you would have this muscle that's continuously contracted, right? So over here, you would have continuous muscle contraction, right? And that too is not good. Um, this continual contraction of the muscle. So that's two ways that you can affect muscles. So now we're going to move on and we're going to go take a look at the excitation contraction coupling. So I just said that these filaments, they slide over each other, right? But now I want to link what we talked about with the actual movement of these fibers for contraction. So how does a nervous excitation couple to the muscle contraction? How does this nervous signal cause muscle contraction, right? We're missing a few steps here. So I talked about up until step 6 here, where I told you that there would be sodium that enters into the sarcoplasm, right, onto this side. So now we're going to continue our story. Once the sodium channels, you're going to have, so when the sarcolemma depolarizes, right, when this area over here where the sodium came in depolarizes, this causes an action potential signal to spread along the length of the sarcolemma. So that electrical wave will go, you see in step 7 here, it travels all on both sides of the muscle, and it really, that electrical wave will keep going through. And then step 8, the depolarization of those electrical signal will travel down this way, down the T tubules, right? Deep into the muscle. It'll actually even go down deep into the myofibrils, right? To the ones even in the middle. And near the terminal cisternae, remember that the T tubules here are sandwiched between two terminal cisternae of the sarcoplasmic reticulum. Sodium influx, here you see this wave, right? You see the action potential coming down. This wave of electrical activity, right? This sodium influx over here will trigger calcium channels, these purple calcium channels on the sarcoplasmic reticulum to open up. And this will cause this big accumulation of calcium in the sarcoplasmic reticulum. There's a high amount. Remember that this is like the storage place for calcium, right? Well, this high amount of calcium, right, will then go and be released into the sarcoplasm all around the myofibrils. So you see that over here, these gates of calcium, they are just releasing all of this calcium into the open of the muscle fiber, right? So that's where we are now. How do actin and myosin interact? Now we'll talk about a little bit the steps. So when a fiber is stimulated, remember I said that the calcium is released from that sarcoplasmic reticulum, and it will go and bathe all of the muscle fiber, there's a lot of calcium stored. It's like opening up the cupboard and all of the stuff falls out, right? So all of this calcium comes out, right? Then calcium will bind onto the molecule that's called troponin. Over here is the actin filament, right? Here's the myosin filament, the thick filament. Calcium actually binds onto this yellow troponin. And this will change the shape of the troponin. So here you have troponin without calcium bound, right? And actually you see that they're separated. This actin fiber is not touching this fiber over here. Here on, you see that now the calcium will bind, and you're going to see what the changes are. So once the calcium binds onto the troponin changes in shape, and the troponin's change in shape moves tropomyosin. So do you see this fibery molecule that is all along the actin filament? This molecule here is called tropomyosin. Do you see over here that tropomyosin is actually blocking the little holes in the blueberries here, right? It's blocking the little holes, but once calcium binds on, the troponin will move the tropomyosin off of the little holes of the blueberries, right? So now you see the holes are exposed. This is because it moves the tropomyosin off the actin active sites. These are called, the little holes here are called the actin active sites. This is where myosin binding will occur. Myosin, this little myosin head, will bind onto these little holes. So now this actin is ready for the myosin to bind, right? So myosin can now bind onto the actin. You see that it binds on now, right? And it forms what we call the cross bridge, because there's like one place over here, another place over here, and this is making a bridge between the two fibers, right? Between the actin and myosin, we have a bridge, a cross bridge. We've crossed the bridge over here. So now the actin and the myosin are connected, and between these two filaments. And the myosin, this molecule here, can now perform what we call the power stroke by pivoting its head. So now it can move the fiber, right? So it binds and it moves the fiber and it pulls on it, right? So this little head here will pivot back and forth and move the actin fiber. So I know there's a lot of names here, but I'll give you a little bit of some study tips. So tropomyosin, right, this little blocker of the heads, this says no to myosin, right? The tropomyosin says no to myosin. Troponin, right? This sounds like opening, right? Opens the binding sites. So it allows the sites to be open. How does it do that? By moving the tropomyosin. So step one, calcium binds to the troponin. Step two, troponin changes its conformation and moves the tropomyosin out of the way. Step three, the myosin heads can now access the actin active sites on this actin molecule, right? So we're all set. All right. So how do actin, so now the other way around, when there's no calcium that floods into the sarcoplasm, so usually when there's a muscle contraction, remember all of these steps, the action potential comes down, we have the release of the vesicles, action potential, this will allow calcium to be released from the SR, right? And then, right, this causes a contraction, so it causes all of these events we had. But then, right, what happens when there's no calcium that floods into the sarcoplasm? What happens if there's no more nervous signal coming down, no more calcium released from the SR? Well, when a muscle fiber is not stimulated by a neuron, right, calcium is not released from the sarcoplasmic reticulum, and therefore no calcium is found in the sarcoplasm. You have none of it there. There is no calcium to bind onto the troponin, right? No calcium binds onto here. Troponin will not move, meaning that this tropomyosin here stays and blocks the actin active sites. the myosin binding sites, right? That's found on this actin molecule. Myosin cannot bind. You see here it's not binding onto these actin molecules, and it will not be able to form a cross bridge, and therefore it won't be able to pull on it and do the power stroke to contract the actin inwards, right? So all of this means that there won't be contraction, right? So here's a question. Imagine whether the myosin binding sites on the actin will be more likely to be exposed or blocked in the following two scenarios. Likewise, predict whether you would expect overall myosin binding to increase or decrease in each scenario. So an image is shown for reference over here. The first situation is there is no tropomyosin present on the actin filaments. So over here, I want you to talk about the myosin binding sites. Is binding increased or decreased, right? And then over here, the calcium binding sites on the troponin molecule are non-functional. What would happen to the myosin binding sites, and would binding increase or decrease, okay? Alrighty. So pause the video and take a crack at this. When you're ready, unpause and we will go through this. So there's no tropomyosin present on the actin filament. Tropomyosin, remember, tropomyosin says no to the myosin. That's this green filament that's blocking the holes for myosin to be on, right? It's saying no to myosin. So if there's no tropomyosin present, right? What happens to the myosin binding sites? They are exposed, right? They are exposed, and or you could say they're available to bind to. They're open, not really open, but they're exposed, right? And binding will increase, right? This is our little actin filament. This myosin filament, if this green was not there, it would very likely bind onto these little active sites, right? Because now we have them present. So this would increase the amount of binding. Let's say the calcium binding sites on the troponin molecules are non-functional. So over here on the troponin, right, this is the molecule responsible for opening, right? So it pulls on the green, moves it out the way so that the myosin can then bind. So let's say these sites over here were blocked and calcium could not bind. Calcium usually binds onto these. Let's say this couldn't happen, right? The myosin binding sites would be constantly covered because there's no signal to move the green tropomyosin out the way, right? The troponin isn't working, so calcium can't bind to the troponin, so the troponin can't move the green tropomyosin. Therefore, the myosin binding sites will always be covered, and binding will decrease, right? This myosin over here will not be able to go and bind because they're always covered, right? So that's going to be our situation here. Now we're going to talk about our cross bridge cycle. So we're going to piece everything together, our last part. We need to talk about what the power stroke is first. The power stroke is myosin heads, right? These are the little heads on the myosin molecule. These are hinges, and they flex back and forth, okay? And this flex back and forth, right, when bound to the active site on actin. So this is important. When the myosin heads are on the actin active sites, when they are bound onto the little holes in the blueberries here, right, that's when they can move back and forth, okay? It's kind of like a paddle of a kayak, right? It's only when you're in the water that you move your paddle back and forth, right? So this is the same situation. You're gonna go onto the actin, and then you see that its head moves, right? And its head was like this first, then it moved and it kind of it will bend and flex this way. So this whole actin filament will be pulled inwards, right? Just like a kayak, right? You take your paddle, you put it in, and then you move, and your boat will move forward, right? This is kind of like the same concept. So over here in this position, right, we say that the myosin head is in its high energy conformation. In your textbook, they call this the cocked conformation because it is in a position that is in this high energy, ready to paddle, right? This is what I think of in the kayak, right? You're at the front of the water, ready to have this. You have this high energy position, ready to paddle and row. So this is the ready to row position, and then once it's in this position, right, then the power stroke occurs and actin is moved towards the M line. So then you actually paddle back, right? You see that there was this push and the head moved this way, and the whole actin filament went to the left towards the M line. And now in this position, we say that the myosin head is no longer in the high energy cocked position. It is now in the low energy conformation. This is the way the you're actually not in any use in this position anymore. Just like in the kayak, at this end, you need to go and put yourself back into the high energy position, back at the front, right, to then push. So this is the low energy conformation of myosin. This is when it's done rowing. What is the cross bridge cycle, right? So now I'm going to go through the steps of how this occurs. So the contraction cycle involves a series of interrelated steps. It begins with the arrival of calcium ions within the zone of overlap in the sarcomere. So here in purple, you see the myosin, you see another myosin on top. Here's the actin in between, right? And in the first step, right, you have the arrival of calcium. Remember where calcium came from? The sarcoplasmic reticulum. It released all of that calcium, right? So calcium arrives, and calcium binds onto, over here, troponin, right? So you have this troponin molecule here, these little orange molecules. So the calcium binds to troponin, and the troponin molecule changes the position and rolls the tropomyosin out of the way. Do you see how this tropomyosin, this green tropomyosin, blocked all the sites here? Well, now that the calcium bound to the troponin moved the tropomyosin, and now you see these active sites in yellow here, right, on the actin. And this allows for the interaction between the myosin heads here and here and here with this active site of the actin, right? So now there will be a cross bridge formation, right? Once the actin active sites are exposed, this is the energized myosin head will be able to bind onto them and form what we call the cross bridge, right? Step 4, the myosin head pivots. After the cross bridge forms, right? Once over here you had the binding of this little myosin head onto it, the energy stored in the resting state is released as the myosin head pivots towards the M line. So you see here is going to be the middle, the M line, somewhere down this way, and all of them will move in this direction, right? So all of them will move that actin filament. In this case, it's to the left because the M line is closer to the left. And this action is called the power stroke, right? When it occurs, the unbound, there was some ADP and phosphate here. So here you see ADP and phosphate that's in this little myosin head over here, right? Well, the way that it allows it to go and do the power stroke, that ADP and phosphate, when it gets released, it tells the myosin head to move and to pivot in this direction. So that's when ADP and phosphate is released. Then, once you're over here, right, well, you're not going to stay stuck and move your head back. right? It's as if I took my paddle, I put it in the water, I moved the paddle, and then I kept it in the water and I moved it back. My boat would be going back and forth, right? You know, when you kayak, you put your paddle in the water, you push in the water, you lift your paddle out the water, you move it back into the high energy position, and you put it back in the water. And then you push again. So now, once we've pivoted, we have to detach, right? We need to get the paddle out of the water and bring it back into its position. So here, when another ATP will bind onto this myosin to re-energize it, and that will cause the detachment of the cross bridge. So when ATP binds, it detaches from the cross bridge. It binds to the myosin head, and the link between the myosin head and the active sites on the actin is broken. The active site is now exposed and able to form another cross bridge, right? So everything gets released. And then there's myosin reactivation. So myosin reactivation occurs when the free myosin head splits ATP to ADP. Remember, using energy, using ATP means to break off a phosphate from ATP. Well, that happens when this ATP here breaks off a phosphate, and you see the difference here. It repositions the head back, right, into this high energy cocked position. Here you see this little guy is in the low energy and ATP binds, and then it is back into this high energy position. And now you have ADP because you took off a phosphate, right? So myosin reactivation occurs when the free myosin head splits ATP into ADP and phosphate, and the energy released is used to recock the myosin head back into its high energy position. Now it's ready to rebind and push again, right? So again, I'm just going to go over again. In this case, right, your oar is above the water in front of you. This position, you put your oar down into the water, right? Number 4, once in the water, you push the oar back and your boat will move forward. So here's where the actin actually moves forward, right? Then in this position, you lift your oar out of the water. And in this position, you reactivate by bringing your oar back to the front of the boat so that you could put it back down again. And this occurs over and over again. We're going to watch a little video to take a look at this.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
All right, so in this question, it says the events of the cross-bridge cycle as they relate to actin and myosin are numbered in order below. Separately, the events as they relate to ATP are labeled ABC, but not necessarily in the correct order. Match the steps as they relate to actin and myosin to the steps as they relate to the ATP by matching the letters together. Note that not all numbered steps correspond to a step as it relates to ATP. So, number 1, myosin heads bind to actin. The power stroke, myosin head release the actin, or myosin heads move to the cocked position. So pause the video, take an attempt first, and when you're ready, you can continue. Now, myosin head binds to actin. Let's go back over here. When the myosin head bound to actin, right? Do you see this is the step that this happened, the cross-bridge formation. Nothing changed here in terms of this is ADP and phosphate in the myosin head. This is ADP and phosphate. So nothing happened here, right? So if I go to this question, this one, actually nothing happens to the ATP, right? The power stroke, remember what power stroke means? It means that when the myosin head actually pivots, right, called the power stroke, so it's actually moving the myosin head, and you see here what's exiting out is ADP and phosphate. So you have the exiting from the myosin head of ADP and phosphate. That's what caused the power stroke. So ADP and phosphate are released from the myosin. I believe this would be C over here, right? So now we got this one. Myosin head releases to actin, right? So let's go back here. Once the power stroke is done, the myosin head releases from the actin and it moves away. Here it's attached, here it's not. So when does that happen? That happens when a new ATP gets brought into the myosin head. So over here, ATP binds to the myosin head, right? So this is A. And then the myosin head moves to the cocked position. Over here, once the ATP is inside, right? The ATP that's inside gets converted and broken down, hydrolyzed into ADP and phosphate. So it gets broken apart, ADP and phosphate, and it brings it back to the high energy conformation, the cocked position. So over here, we would say the myosin head moves back. That's when ATP is hydrolyzed. Remember, hydrolyzed just means hydro using water, lysis to cut. So we cut the ATP into ADP and phosphate in that step. That's number 4. Okay. So we're pretty much done. I just need to like piece everything together and summarize because it was a lot of steps, right? The link between the generation of an action potential from a neuron here and the sarcolemma, the muscle, and the start of muscle contraction is called the excitation contraction coupling. The nervous system is coupled to the contraction events. They're linked, they're coupled. You need one for the other, right? So I want to go through the steps of this excitation contraction coupling, how it all happens together, right? So step one, acetylcholine. Actually, there's a few more steps between that, but before that, but acetylcholine eventually will be released from the synapse here, right? And bind to its receptors on the sarcolemma over here, generating an action potential. You see this red arrow, it's trying to show that this is an excitation, an action potential, an electrical activity in the membrane of the muscle. Step two, the action potential travels down the sarcolemma and down the T tubules, right? Um, so we go down into the T tubules. Once the action potential reaches the bottom of the T tubules, right, it stimulates this sarcoplasmic reticulum, which is a big storage of calcium, right? It stimulates this big storage of calcium to release their calcium ions. This was waiting to be released, right? Just like that cupboard that's full of clothes, when you open it, it all falls out. It was waiting to be released. It was just stored there. Step four, that calcium, right, will bind onto troponin. We don't see that in this picture, but we know that calcium binds to troponin, found on the periphery of the actin filaments, on these red filaments. That will cause troponin to move the tropomyosin out of the way. We don't see that in this picture, but we know it happens. This allows the active site on actin to bind to the myosin head. So the red and the purple will then bind and they'll form a cross bridge, right? So we know that that cross bridge will allow the myosin heads to pull these actin filaments towards the middle. On this side, they'll pull these actin filaments towards the middle here, so the actin filaments will move, right? That's kind of linking everything together with a bit less detail, but that's the big picture here, right? We went through each of these steps. We went through the neuromuscular junction. We went through the sarcoplasmic reticulum. We went through the calcium binding to troponin and moving the tropomyosin. And we went through how the myosin binds onto the actin to actually move it, right? So each of these are a separate step, but they're linked. They're coupled. How do things end? Well, acetylcholine is broken down, right? Over here, we released a bunch of acetylcholine, and all of a sudden, though, it's broken down and there's no more, right? Acetylcholinesterase will help to break down that acetylcholine. That will end the no more action potential generated over here, no more action potential down the T tubule. So in the sarcoplasmic reticulum, there's no more calcium that's being released. Actually, instead, the calcium will be reabsorbed back into here, right, through an active process. It'll use ATP to reabsorb the calcium into the sarcoplasmic reticulum and store it back. The active sites will be covered because there's going to be no more calcium outside. The active sites will be covered, right? The tropomyosin will return back and cover the actin, right? And because of that, the myosin will not be able to bind onto the actin. Without cross-bridge formation, contraction ends, so myosin will not bind here. And now you see that this red actin is not bound to the purple myosin. And so it'll actually return back to its original position. Remember, there's like these little elasticy fibers, so the actin will return back to its regular place, right? And so here we were contracted, here we are relaxing, all right? So now I want you to attempt this one. The term excitation contraction coupling refers to the events that turn the action potential into a muscle contraction. Put the steps in order here. So I want you to pause the video and try to do this. So the action potential propagates through the muscle fiber. Here we have the calcium binding to troponin. myosin binding on actin, so the myosin binding. Here we have the action potential traveling down the T tubules. Here we have some voltage gated channels open, releasing calcium into the sarcomere. Troponin changes conformation, moving the tropomyosin. So we have a bunch of stuff here, all different orders, right? Now I'll tell you how I'd go and tackle this specific problem over here. So for C, action potential travels down the sarcolemma and T tubules. This had the key term of the action potential and the sarcolemma and the T tubules. Oops. This is happening up over here, right? The T tubules, the sarcolemma, everything else that's mentioned here, right, has to do, we have troponin, that has to do with this step over here, right? So troponin, here it says the myosin binding sites on actin are exposed. That also has to do with the troponin situation. Voltage gated channels open, releasing calcium into the sarcomere. That's this over here, right? Remember the calcium that gets released? So that happens actually a step before the troponin and all of that. So this actually, I'm going to go and put in a different color. Troponin changes conformation and moves the tropomyosin. This has to do with the troponin and the tropomyosin. So I definitely know that C comes first, right? The action potential comes and then blah, 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 and the action potential travels down the sarcolemma and the T tubules. Okay. As I said before, that is like this situation here where it goes and travels down the T tubules. Then the SR situation happens, right? The sarcoplasmic reticulum. So that was the D situation, right? Where calcium gets released from the SR into the sarcomere. Then we have all these green ones. That happens in this case over here, right? When the calcium is released, what happens? And actually it happens, we looked at it in this slide here, right? The troponin, the tropomyosin. So let's go back and kind of look over those options. The first one is calcium binds to troponin. That seems, once the troponin is released here, I think this is very logical. Let's put that first for now. Oops, let's just undo that. Put it in green. So here's A, right? Myosin binding sites on actin are now exposed. So that seems good. So the calcium binds to the troponin. Let's read the other one in case. Troponin changes conformation. So once calcium binds to troponin, what happens? The troponin changes shape and moves the tropomyosin. That's next. Then the myosin binding sites are now exposed, right? So that would be my B. And now the cross bridge can form and the muscle can contract. Okay, so that was what we had for our cross bridge cycle. Now we talk about muscle performance and use of ATP. So now we're done with the main stuff. Now there's just tiny little topics to talk about. So muscle performance. So how is ATP regenerated during muscle activity? So because ATP is the only energy source used directly for contractions, it must be regenerated as fast as it's broken if contraction continues, right? So ATP, it's not like other molecules are being used for the muscle contractions. ATP is the one, right? But we can make ATP in different ways, and it actually happens in different ways depending on what level we're at, how much time has passed for muscle contraction. So we're going to see the first way is through direct phosphorylation of ADP. So you're going to see this ATP being created because ADP is going to be phosphorylated by creatine phosphate. This is found in muscles, okay? That lasts 15 seconds. When you contract a muscle, that'll last 15 seconds. The other way is through anaerobic pathway. Ana means no, aerobic means oxygen, right? So no oxygen when there's not oxygen available. We're going to do this fermentation pathway of glycolysis and lactate. We'll talk about it in a bit. This is going to last about 30 to 40 seconds, okay? And then aerobic means having air, having oxygen. We're going to go through this other crazy pathway where we're going to make a lot of ATP. This is for sustained muscle contraction. If your muscle is contracted for hours, it'll go through this pathway here if oxygen is allowed to be there. So I want us to look at these three different ways that ATP is regenerated. First, the direct phosphorylation by creatine phosphate. So do you see this creatine phosphate? It will turn to creatine, it will lose a phosphate, and it will give that phosphate to ADP so that it could make ATP. So let's take a look at that. At rest, muscles store ATP as creatine phosphate. ATP is too unstable to store and transport around the body. Remember, it has those three phosphates that are negatively charged pushing on each other, right? So actually, it's too unstable to store and to maybe send to the muscles or elsewhere. Therefore, the mitochondria of all cells create their own ATP. Your liver cells make their own ATP. Your brain cells make their own ATP. Your muscle cells make their own ATP. So each cell has an ability, if it has a mitochondria, to make ATP. In muscle cells, however, they're special. They're smart. They have an excess, when they have excess ATP, they'll convert it into their own little storage in their muscle cell in a stable form called creatine phosphate. Remember, I said storing ATP, it degrades quickly, it's unstable. So instead, it stores it. Muscle cells are smart and they store a little extra reserve as creatine phosphate for later use. And this is how they do it. ATP, when you have too much ATP you made, plus creatine, right? Through the enzyme creatine kinase. Kinases are enzymes that add phosphates. So adding a phosphate to creatine, it'll take the phosphate away from ATP, which has triphosphates, 3, and it will put it onto the creatine. So do you see how this ATP turned into ADP? It went from 3 phosphates to 2. It lost a phosphate. Where did that phosphate go? This enzyme put the phosphate onto the creatine and created creatine phosphate. If we just left ATP like that, as I said, it's too unstable, it degrades, it wouldn't stay as a good way to store it, right? It's like when you cook a big batch of something, you're not just going to stick the pot in the fridge. Sometimes you put it in little containers and it stores better, right? Well, same thing in this case, okay? Why do we store it, right? If we have excess ATP, we store it in muscle cells because during activity, creatine phosphate can be converted back to creatine and ATP, and therefore we can have ATP available. Maybe I didn't emphasize this enough, but I want you to realize over here, right? ATP is used in many places. It's used, the most important one we talked about, right, was over here, ATP. You see the ATP molecule. We see that ATP is essential. Every time that little myosin head will detach, it needs an ATP. And during a muscle contraction, one muscle contraction, this happens many, many times. So muscles need constant ATP, right? That's why they have their own glycogen storages there. So I want to just emphasize that again. Muscle contractions require a lot of ATP. So as we begin to exercise vigorously, ATP demands will soar. They will go up. We need ATP, right? And in working muscles, it's consumed within a few twitches. So just a few muscle twitches, you will need more ATP. You will use up all the ATP that you had already. So the creatine phosphate is useful because these storages are converted back to ATP and they could provide energy for about 15 seconds. So this ADP that we generated plus creatine phosphate, right? The creatine kinase will take the phosphate from creatine phosphate and put it onto this ADP molecule to form ATP, right? And therefore now this could be used during the muscle contraction. When the muscle was resting, we didn't need the ATP, so we put it in this form. And when our muscle contracts here, now we need it, we make ATP, and now we have creatine as a byproduct, right? and we can use the ATP now. Interestingly, this is just a little side note. Creatine is slowly converted into a waste product that's called creatinine, which is excreted by the kidneys into our urine. So, creatinine in urine is a good indicator of kidney functioning or how much muscle mass we have. So, that's just a little side note because sometimes in blood tests we look at creatinine. So this happens, right? I want you to remember this direct phosphorylation by creatine phosphate, that happens in the first 15 seconds of a muscle contracting, right? Like think of it now though. Like right now, many of your muscles are contracting, you just don't know about it. Just because you're not bulking your biceps doesn't mean that your muscles aren't contracting, right? You have back muscles that are continuously contracting during the day, so this wouldn't be enough for your those muscles, right? After 15 seconds of contraction, we need another source, right? So as creatine phosphate is exhausted after 15 seconds, ATP generation occurs by an anaerobic pathway. So over here, this is your anaerobic pathway. What is our energy source? Our energy source is the breakdown. Remember, to get energy to make ATP, we need to break down bonds and use that energy, right? It's like a fire. To make a fire and make heat energy, we need to burn wood to break the bonds of wood to make heat, right? While here, to get energy, ATP, we need to break bonds of food, right? So here in this case, we break down glucose. There's two places where we have glucose. We break down glucose from glycogen stored in muscle. Actually, our bodies, remember I told you glycogen? We store glycogen in muscles and actually in the liver. We store it in the muscles, glycogen, this long polymer, right, of glucose, in for the time when we're going to need it, right? It's like when you go to a grocery store. You don't just buy one meal every day at the grocery store. You buy it for the week and you store it in the fridge and you slowly take it. Well, this is what happens. We take our glycogen storages. When we start using our muscles a lot for more than 15 seconds, we start taking those glycogen storages and we break them down. We also break down glucose from the blood. So, sometimes you'll eat and you'll just have low glucose around. Well, actually, you'll break that down too, right? Um, and this is called glycolysis. This is the breakdown of bonds in glucose, and it uses the energy to make 2 ATP. So when you take this glucose, you undergo glycolysis. This is a process where you take glucose and you make this pyruvate smaller molecule. You will make ATP. You break bonds down in glucose and you make ATP. Lactate is a byproduct that's made and released into the blood at the end of this process. This is going to be found in your blood. And I want you to know that in this process, this is anaerobic, right? No oxygen is needed. It's not required. What do we make as products? We make 2 ATPs per 1 glucose, and we make some lactate. This energy will last 30 to 40 seconds since there's not much ATP produced here, right? So actually, we went from 15 seconds to 30 seconds. I know that you can lift a box up for more than 30 seconds, right? So what do we do if we need energy for a longer term, right? Well, this is aerobic respiration. Since anaerobic pathways don't create much ATP, the aerobic pathway requiring oxygen will kick in to sustain a very prolonged muscle activity for hours maybe, okay? So what is the energy source for this? As exercise begins, it first comes from all those glycogen storages in our muscles. That's the main fuel, right? Shortly after, major sources of fuel will come from the breakdown of glucose in the blood. We will have pyruvate from glycolysis. Glycolysis still happens and we still make pyruvate. So pyruvate will go to the mitochondria here. Lactate from anaerobic processes will also come in and feed into this process. And free fatty acids that are found in the blood will enter into the mitochondria and do this. So this is in the beginning, shortly after. After about 30 minutes, actually, you're going to get rid of all the fatty acids in your blood, and you're not going to have any more glycogen. You're not going to have any more fatty acids in the blood. So you're going to go to your fat cells, the triglycerides, and you're going to break down the triglycerides to make fatty acids in the blood. So you're actually going to start breaking down your fat, right? That's why exercise, if you exercise for a long time, you'll start breaking fat down. So that will be the major fuel. And so the mechanism of this is all these nutrients, right? Things that we eat, glucose, fatty acids, amino acids, they feed in to this chain, and you will undergo glycolysis sometimes first to make some ATP, but then the rest of them will enter into this aerobic process that occurs in the mitochondria, and this produces a large amount of ATP. A lot. This made 2, this makes 32 ATP. So this is very good for muscles to undergo aerobic respiration. They're going to get a lot of energy and they'll be able to sustain for a long time. This requires oxygen, however, which is why when we do physical activity, we start breathing heavier because our oxygen demands increase because we're using it a lot for this. And we produce 32 ATPs for every 1 glucose, right? It's different for fatty acids, but we don't need to worry about that. We also, though, in this process make a lot of CO2, which is why we start to, exhale a lot more too, because we make this byproduct that we need to get rid of, an H2O. So the duration of this is actually hours. This can allow you to contract your muscle for hours, right? This is a reason why we need to breathe. We need oxygen for this process over here, right? Our cells need oxygen to do this and make ATP. Last two little topics, muscle fatigue. So what is muscle fatigue? It's the inability to contract, even though the muscle is receiving a stimuli, even though your brain is telling your muscle to contract, right? For some reason, sometimes the muscle doesn't do it. I could imagine you have a huge load that you're carrying and then your muscles give out and you drop it, right? Why does that happen? Well, muscle fatigue, it actually is important because it prevents complete depletion of ATP. If we fully got rid of ATP and we no longer could sustain it, then that would cause death of our cells. Our cells would no longer have, our muscle cells wouldn't have any ATP, and therefore they wouldn't be able to do just their basic functions, never mind contraction, but just their basic functions. So we don't want that to happen, so muscle fatigue occurs. And the science behind this is not super clear, but here are some of the changes involved in muscle fatigue. Why is it that our muscles fatigue? First of all, the ionic imbalances. Once the levels of potassium, sodium, calcium all change and move around, right? We saw that calcium moves around, sodium moves around. If that kind of gets off of whack, then the membrane potentials of muscle cells will change, and that may lead to help cause muscle fatigue and stopping us from contracting, right? Even though we're saying keep holding it, right? That signal might allow our muscle to fatigue and stop. Secondly, inorganic phosphates. This inorganic phosphate from creatine phosphate and ATP breakdown may interfere with calcium release from the sarcoplasmic reticulum. So this is that phosphate, right, from the breakdown might have some kind of effect on release of the sarcoplasmic reticulum. Another studies show decreased ATP goes in tune with increased magnesium levels. As ATP levels drop, magnesium increases, and this can interfere with voltage sensitive T-tubule proteins. That might be one of the other reasons why your muscle stops. And lastly, when you have decreased glycogen, this is highly correlated to muscle fatigue. So this seems to be another important factor to keep your muscles when you start having lower and lower glycogen, then your muscles, this might be one of the mechanisms that explains muscle fatigue. What happens after vigorous muscle exercise, whether or not fatigue occurs, right? Vigorous exercise will alter a muscle's chemistry dramatically. So, for a muscle to return to its pre exercise state, the following need to occur. Oxygen reserves need to be replenished, right? Remember over here, we need oxygen to undergo this process. So if we want to continue, so after we're done this vigorous exercise, right, oxygen reserves need to be replenished. Even after you're done, going on your run and using your muscles a lot, you're still going to be breathing heavy to bring in more oxygen. Glycogen storages must be replaced. This takes a bit of time, right? You need to rebuild your glycogen storages that were in the muscle. You just emptied out your cupboard, plus you were constantly bringing stuff from the grocery store, right? So you've emptied out everything. You need to go back and replace these storages. ATP and creatine phosphate reserves, they need to be resynthesized and be brought back after exercise. So this actually shows that even after you're done your exercise, you're still working to remake these things. You're still using energy to make these things, right? So even after exercise, you're still working, right? Even though you're tired like this. So what is excess post-exercise oxygen consumption? All these three replenishing steps above, they require oxygen uptake and aerobic metabolism. They require this here, right, to occur even after exercise. So once exercise stops, repayment needs to begin. So the extra amount of oxygen that the body must take in for these steps to be replenished, right? And other restorative processes. This is called the excess post exercise oxygen consumption. Basically, it's called the oxygen debt. You are in debt. You used up all these storages and now you are in debt. You need to bring in more oxygen and you need to actually make more ATP to do these things, right? So, EPOC is basically how much work are you doing even after you've done exercise, right? You're actually still working out after your workout, even though you don't know about it. So this epoch can last for hours to days depending on the type of exercise and the intensity. So it's fun that you've stopped your exercise, but you're still paying off for that exercise, right? And lastly, skeletal muscle response to exercise. I just want to talk about two types of exercises. There is aerobic exercises or endurance exercises. This is like running, fast walking, swimming, biking, right? This leads to several recognizable changes in skeletal muscle. Um, Increased number of capillaries around the muscle fibers, increased number of mitochondria within the muscle fibers. This is what's going to happen during this type of exercise, right? Endurance, trying to see how far can you go, right? The changes result in more efficient muscle metabolism and in greater endurance, in greater strength, resistance to fatigue, right? But then there are these other types of exercises called resistance exercises. These are typically anaerobic, and it pits muscle strength over stamina. Strength is what is most important compared to how long you can do it for. For example, weightlifting, it's a quick activity. You lift these very heavy weights, but it's quick up and down, right? As opposed to maybe a lighter weight for more repetitions, right? This is more about a high weight just done a few times quickly, right? And this will lead to something different. It leads to increased individual muscle fiber size, right? So actually muscle hypertrophy, you actually get a bigger muscle fiber, bigger muscle cell. It increases the myofilaments and myofibrils in that, and that's why the muscle fiber gets bigger. It increases your glycogen storages and it increases the connective tissue between the muscles. So this is a different response compared to an aerobic exercise. And they all have, both their pros at what they're doing, but they're doing it in a very different way. Okay. And that is all for this lecture.